And we are open for questions. I don't have anything through the computer, so I'll go ahead and Josh, you have a question?
So thanks. These were all terrific. And my questions go back to money because we've talked about that a fair amount. And John did not talk, for example, about how much it would cost to do any of this work. So I'm wondering if you can touch on that. And then I also have a question for you folks who address COXI fairly frequently, and that is from the medical legal standpoint. We've heard a lot about how much it costs to take care of somebody through the medical practice, but we also know that there are millions of dollars paid out for workman's comp. We know there are millions of dollars paid out for the prison infections, and there are also millions of dollars paid out for delayed diagnoses. And Farbe, you really shared beautifully in your slides how people are waiting a month to almost a year. And that delayed diagnosis is one of the very common sets of medical malpractice. So I'm wondering if you can talk about money.
So, John, do you want to talk, John Rex, talk about how much? John Rex.
Yeah.
Rex. Yeah, that's why I said John Rex.
Yeah. So what I'll say is that my rule of thumb for the per patient cost for a complex study is $100,000. And there are lots of things that go into that, but that's the whole study cost. That's the running the study site, that collecting all the data, doing the patient visits, paying for all the lab tests, and it will obviously be influenced by how much is in hospital, how much is out of the hospital, a bunch of things that go into it. But at the end of the day, my experience is you spend about $100,000. You don't spend $100,000 on each individual, but it's all the stuff you got to wrap around them and around the data collection processes. That's what it ends up costing, is in that range. So it depends on how big the study is. You think you need 100 people, you're probably, that's $10 million people. $10 million.
I'm going to take my moderator prerogative and ask John Rex. So the problem we have as clinicians, as we've pointed out, we only have 2 drugs with FDA approval. So all other drugs we need to get and that we've used are not FDA approved. And this creates an enormous problem in clinical practice because we have to get prior approval and the drugs cost a lot of money. And this is limiting. So obviously most of us here, at least, and certainly me, would like to see if we got a new drug that it would be FDA approved. So what are your opinions about how best, what would you propose to get a new a drug FDA approved for a COXI indication?
Well, I mean, I told you that on my slides. And I think that...
Well, I want you to get down to, well, you gave us options. So what would you say is the one best option to pursue?
It depends on the qualities of your agent and how comfortable you are with the safety profile. What else you've been able to do with it before you come to COXI land. If you're able to study some other fungus first, you might be able to back into it. And I think that the ones where you're most likely to show a strong effect were the 3rd and 4th categories that I just discussed. But those are also diseases that I wouldn't enter lightly. I would want some comfort that the dose we had chosen was actually really going to do something before I enter a study like that. But once you got to a study like that, I think it would really stand up and shout if you could deliver a demonstration of superior response when you have somebody who's got final involvement or something. I think that would really be a very strong result because you'd be working in the harder form of the disease.
Right. Any other questions?
I can't answer about the malpractice issue, but I have an answer about the laboratory testing. Actually, in Arizona, our patients can go to SonoRequest lab and request the lab screening for valley fever without a physician's prescription or order. And that costs, even without insurance, about $30-some, if I'm not mistaken. So it's really not a very expensive test. The question is, if that is negative, then it remains negative, and if it is positive, it's reported to the patient's primary care physician, then that person has to, manage the patient accordingly.
There's some questions is that Mitsuru?
Thank you. Mitsuru Toda, CDC. Excellent talk. Thank you. This is a question for Dr. Thompson. For the TDM and susceptibility testing, what do you think are the biggest barriers for lack of uptick? And do you see any role that public health could play in that space?
I think so. I do have to admit my bias that I see a very end of the bell curve spectrum of coccidioidomycosis cases. I tend to only see ones that I see almost no primary pulmonary disease other than those over a telemedicine clinic. So the patients I am referred are usually have failed multiple options already. So I fully admit that is a bias and a lens through how I view these patients. But I see a lot of elevated MICs, particularly in that population. The isolates that have been sent to Texas they may be a biased group, right? So there was some reason that someone sent susceptibility testing. That's not something we're used to doing with Coxie. So it would be nice to have some soil isolates, and I know Bridget has access to those, and so does some other people, and do susceptibility testing on soil isolates to make sure they also have higher MICs. Because then I think we can really drive home the point that, look, these just are, have a higher de novo MICs to fluconazole at least. I think that would cinch that up pretty well, at least for me, to have the data from the soil isolates also. I think, for some reason that the lessons we've learned in other areas of mycology have not been applied to Coxie. If you failed primary treatment for aspergillosis, we're very quick to look for resistance. If you fail treatment in Candida, you look for resistance. I mean, in Canada, we look up front, right? Before, we often have that back within 48 hours. So I think we've been a little slow to adopt a similar philosophy with Coxie. I think part of that has been the difficulty in obtaining some of these other drugs for the reasons that Neil outlined. A lot of times the insurance just says, no, you can't use them, and we're kind of stuck. So I think that's sort of the message. For therapeutic drug monitoring, I think that's also really going to depend on your comfort level with these drugs by what type of institution you work at. If you work in an institution with a heavy leukemic and BMT population, you're probably used to using these other drugs and are used to checking therapeutic drug monitoring. If you don't, you may not use those other azoles quite as often and then just assume that the levels are going to be normal based on that you don't check fluconazole levels routinely. So I think that there's just a bias in all of our practice patterns, and that's probably just something we need to educate more people on.
Okay, Mark?
Hi, George. I had a question about measuring drug susceptibility. So we know that there's obviously some mutations that lead to drug resistance. But my understanding that measuring drug levels for Coxie are done by looking at hyphal growth in tubes. And I'm wondering why someone hasn't developed a spherule test for that, because there may be differences based on the very different wall structure and growth phase of the two forms.
Yeah, that's a great question. I mean, I think that the UCSF group has shown some nice data for being able to look at these in the SE phase. I'm only aware of one isolate that's ever been looked at in the mycelial and the spherular phase, and the MICs were the same. That's one isolate, though, so we definitely need a lot more work in that. I mean, we have no CLSI breakpoints for Coxie. We're not even close to being able to propose that.
Okay, any other questions? Rob?
So as a valley fever patient who's failed most of the drugs that Dr. Thompson talked about, and hearing the difficulties that Dr. Rex talked about, so what is the path forward? I mean, and that's the key thing that patients want to know. I mean, what are the opportunities for, and are there currently any opportunities or thoughts of opportunities to make this path forward easier? I know it's a very vague question, but I would love to see some hope out of this.
John Rex, that's probably directed to you.
Yeah, Rob, that's the conversation I was pointing at in my final slide. The regulatory statutory wording is drug approval is based on, memorize the following words: substantial evidence of efficacy based on adequate and well-controlled clinical investigations. Now, nowhere in there does it say P less than 0.05. Nowhere in there does it define each one of those adjectives. They're defined sort of in practice, was what we have always done. And the agency has a lot of, as they often say, we have regulatory flexibility to think scientifically and to actually, you want the data to be valid, you want it to have some meaning, but there's an ability to flex based on the constraints of the situation. And that to me is the cool thing about the LPAD language. The LPAD made it really clear and gave the agency some lovely language to use when talking about the idea of stretching for an unusual infection to work to do something unusual a little bit different. I just let me point at an example, which is a drug for highly resistant TB, a drug called protominid, which was approved based on 100 patients worth of open label data versus some historical controls. It's basically approved because the response they saw when they put people on protominid, people had failed everything else. What the response they saw was unexpected. It was really, really unexpected. And it's that kind of a thing that I think we need to start to explore with some of these really low frequency, difficult infections, because you're just not going to be able to study. Cocci is reasonably common in your part of the world, but it's not like it's not everywhere. And the number of folks who have cocci meningitis is it's not a huge number. So how are you going to do that study? I think I think that conversation is happening. That was the reason for the FDA's workshop, was to begin to have that conversation. And the best way to force it is to keep poking at it, which is why it's nice to have the conversation today.
Okay, any other questions? Yes?
Hi, this is Gail Sondermeyer-Cooksey from the California Department of Public Health. And Fariba, I wanted to thank you for talking about underdiagnosis and delayed diagnosis. And my question is kind of the next step, is if we get doctors to test issues, if you could comment on issues with interpretation of those tests and the results. And I think we just worked on a new national case definition. And essentially in Arizona and California, any positive is a positive from a surveillance standpoint. But we see a number of people in our data who are only coming in with either an EIA or an IgG positive. And either they don't have titers or their titers are 1 to 2 or lower. And the question of, is that what's causing their disease? And is it potentially doctors misinterpreting those results?
Well, at least in our practice, when any of those tests are positive, I consider that patient have cocci. Now, how do we follow these patients is a different story. If they are in that 40% mildly symptomatic, I usually don't treat. I usually follow these patients. I repeat the test. I'll examine them fully every 2 or 3 months. And they have my number. They always call me. They literally have my cell number. So I'm in close communication with these patients all the time. And if anything changes, I always ask them to call me right away. I see them right away. If we need to image something because something doesn't look right, or there is this pain that was not there, or there's a headache, we immediately pay attention to that. The point is that with the test, our test is about, we use this IMI in our institution, it's 46% sensitive. I don't only rely on negative tests to say patient doesn't have it. And if they do clinically present as cocci, I do follow them very closely.
Okay, any other questions? Well, I want to thank our speakers very much. Excellent. Now, GR asked me as the last moderator to summarize and adjourn. And so we'll be here another hour while I work on that. No, I want to thank all the speakers and all the questioners. This was a very vigorous discussion. We went over just a massive amount of information, and we'll do more tomorrow.